Hello, my name is Fiona, and for my biology honors project, I will be outlining the two main DNA repair pathways that can address double-stranded breaks in DNA and potentially be leveraged to introduce new mutations. It is important to note that this is a general overview of the subject. However, some foundational biology knowledge, such as the function of DNA polymerase and DNA ligase, is assumed. So, what is a double-stranded break, or DSB? That is a great question. DNA, as shown in the drawing, is found in a double helix with adjacent bases following the Watson-Crick base pairing rules. Double-stranded breaks, often referred to as DSBs, are mutations that occur as the phosphate backbones on both sides of the double helix are broken simultaneously. This break on both strands can be a problem because DNA polymerase cannot as easily use one strand as a template for the synthesis of the other. So what causes these double-strand breaks? DSBs can be caused by a variety of factors, including radiation such as UV damage, chemical mutagens, and even natural metabolic processes. The question then are, why are double-strand breaks so bad? This is because they can result in lasting changes on the chromosomes, such as rearrangements and inversions, as well as insertions and deletions to the sequence, all of which can lead to harmful phenotypic effects. Without repair, these double-strand breaks can also lead to cell death. This is why the native DNA pathways that I will be outlining are so important. The first of the two pathways that I will be discussing is non-homologous end joining, which is in simple terms the piecing back together of a DNA strand without a repair template. In the image, you can see an example of a double-strand break. The first step is that nucleotides at the break site are often lost in a process called resection, which creates single-stranded overhangs. The double-strand break is then recognized by end-binding proteins, such as the Ku proteins in mammalian cells. These end-binding proteins serve as recruiter proteins to recruit additional proteins which differ by organism to create a cross-bridge which makes sure that the DNA does not separate in the process. The function of these proteins can differ case by case and by the type of break. These proteins may in some cases facilitate the digestion of more DNA material and end processing, or in some cases DNA polymerase may insert nucleotides. Generally, the ends are made compatible and the strand is ligated again by DNA ligase. This process results in a DNA strand without a break, but with the potential of sequence insertions or deletions, if errors have occurred during the process. One advantage of non-homologous end joining is that it can happen at any point in the cell cycle. However, its disadvantages is that it's error-prone and can result in small deletions in the repair region or insertions, which can have an overall effect if it results in a frame shift or early stop codon. Next, I will be giving an overview of homology-directed repair, also known as homologous recombination, which occurs when a homologous DNA strand, usually from a sister chromatid, is used to repair a double-strand break. Take, for example, a broken DNA strand in a cell in the S or G2 phase. A sister chromatid of our broken strand is recognized as it has a region of homology. First, the DNA at the break site is digested to create overhangs. Next, a strand exchange must occur between broken and unbroken sister chromatids, where the resected end must find, synapse with, and intertwine with the homologous DNA strand to be ready for synthesis. The unbroken strand is then used as a template by which DNA polymerase synthesizes DNA through designated base pairing. The crisscross strands are then resolved and religated to produce unbroken, separate chromatids. An advantage of homology-directed repair is that it is less error-prone because the repair templates from the sister chromatid is genetically identical. However, sister chromatids are only available in the S and G2 phase of the cell in eukaryotes, or directly following DNA replication in bacteria. Additionally, this is a slower process which can be a problem when DNA must be repaired quickly. So the big question then is, why is this important and how can we use this knowledge? The first is through purposeful mutagenesis. Since we know that non-homologous end joining can introduce insertions and deletions into our sequence, we can incur breaks either randomly through mutagenesis or non-randomly through CRISPR. The introduction of just a single nucleotide could knock out a gene or change the overall function. This allows us to make mutant libraries, all with different mutations and potentially varying phenotypic effects. The next is gene introduction. Because we know how the HDR pathway functions, we can provide DNA with homology to a certain region. Then, if we incur a cut via CRISPR, our organism will use this template with homologous arms and integrate the new gene or sequence into its native genome. This opens up a plethora of possibilities to us as scientists, not limited to bioremediation, gene therapies such as diseased allele knockouts, pesticide and herbicide resistance for growing populations, discovery of antibiotics, among many.